Can PBPK model provide confidence? Well, here we try to answer three questions. First of all, are PBPK models actually sloppy? Secondly, if so, how can you predict with confidence? And thirdly, which open project is worth doing? Let's take a look at the PBPK models. On the left-hand side, you have a depiction for the whole-body PBPK model, where organs are connected by venous blood as well as the arterial blood. On the right-hand side, you have different types of parameters going into this model. Broadly speaking, there are two different types of parameters. The organism parameters, which characterizes the organ volumes, surface areas, and so on. And the drug parameters, such as permeability and partition coefficients. Sometimes, some of these parameters can be experimentally measured, but other times they can't. So when they are not, you will need to use prediction models utilizing different type of chemical physical properties to predict the permeability and partition coefficients. So here's the problem. If you use different prediction models to predict the values of permeability and partition coefficients, you may end up with different prediction outcomes. So in the case of seal filling in different tissues, here you can see that the prediction are indeed fairly variable. Also, the model complexity actually rises rapidly with the number of organs, which oftentimes requires unknown parameters, and you need to answer the question which parameter actually matters the most for the PKs of interest. This essentially boils down to the question, what is the appropriate level of details that you need to retain in such a model? First of all, you need to define the objectives. What do you want to predict and how accurate that prediction needs to be? And secondly, this is often a data-driven decision, as you will see in our example. So the benefits of simplicity really is that you should retain only one main hypothesis to test, and this gives you clear results. And also such a results should be easy and clear to communicate. Let's take a look at the example of vitamin D modeling. Vitamin D deficiency is prevalent throughout the world. In the UK, about 1 in 3 Britons are deficient in vitamin D in spring. When we started to look at this problem back in 2018, the pharmacokinetics profile of vitamin D appeared to be unpredictable. First of all, you have extremely variable clinical PK. And secondly, there was a lot of debate about the doses should be given to the patients. And hence, these clinical trials are conducted using different doses. So the question really is, when you start dosing a patient at a particular dose, when do you need to take a blood sample to confirm sufficiency? So we found these data to characterize the pharmacokinetics of vitamin D as well as 25-hydroxyvitamin D after dosing. You've got data for vitamin D after single dose as well as daily dosing. You have data for the 25-hydroxyvitamin D after single dose and daily dosing. Here you can see that for each trial arm, the baseline varies, the dose is very different, and there are a whole bunch of other factors that are different. Hence, the million-dollar question is it possible to make sense of these with a concise model? So this is how we model the pharmacokinetics of vitamin D. Upon oral administration, vitamin D enters the GI tract and ended up in the liver. On the other hand, you have endogenous vitamin D synthesis, and those vitamin D arrive at the liver as well. Some of the vitamin D molecules enter the bloodstream via the venous blood and the arterial blood, and we grouped all the other compartments together into one compartment, which we call the rest of the body. About 1 in 3 molecules of vitamin D will metabolize into the 25-hydroxyvitamin D. and the other two molecules will become inactive metabolite. So the same thing for the 25-hydroxyvitamin D. It enters the bloodstream and goes through the entire body. And finally, the 25-hydroxyvitamin D is cleared in the liver. Here we make only one main hypothesis, which is the endogenous vitamin D synthesis rate is different from arm to arm, and we kept all the other parameters the same. So this model is simple. It is simple in structure because all non-eliminating organs will merge together. And also it is simple in parameters. We have a single set of parameter values to fit all the data. And here are the organism parameters that were taken from a previous publication. So here are the data that we use for training and testing this model. We have 13 arms of vitamin D observations and 43 arms of 25-hydroxyvitamin D observations for training the model. And to test the model, we had 83 arms of 25-hydroxyvitamin D under the daily dosing, as well as 16 arms of 25-hydroxyvitamin D under large single doses. As far as the 25-hydroxyvitamin D is concerned, you can see that the dose range that we use for the testing greatly exceeds of the dose range for the training, and this constitutes a relatively hard test for the model to pass. How well do we do? Here we plotted the model predictions on the horizontal axis and the observations on the vertical axis. Here you can see that the model predictions are in good agreement with the observations for the training data. And then for the testing data, the model agreement was good as well. It only made slight over prediction for extremely high single doses. Hence, we can conclude that the model made accurate predictions for most daily dosing as well as the single dose regimens. Here are some of the arms that we used for testing model accuracy, and as you can see, the model make very good predictions. So what we learned from this model is that patient compliance is key. For a patient suffering from severe deficiency at a very low baseline, if you use a dose of 2000 IU per day, it takes a fairly long time for this patient to arrive at a new pharmacokinetics steady state. However, once you stop dosing, the 25-hydroxyvitamin D levels drops precipitously over time. And here's a bit more information about this model. So here's a summary of the lessons that we learned. The data come from a very diverse range of population with a very wide range of doses being applied. And the model is really simple. It is simple in both structure and parameters. And the model only represents one main hypothesis, which is each arm's unique endogenous vitamin D synthesis rate explains the data variability. And you have only one single set of parameter values for the rest of the parameters. For the results, while we made accurate predictions for most of the test data, it made slight over prediction for extremely high single doses, and the main hypothesis here is therefore strongly supported. And lesson learned, mainly simplicity is key, because that allows you to know what exactly you are testing. For good modeling practice, first of all, you want to use all accessible data from various sources and various conditions. And secondly, when you develop a model, you want to keep it as simple as possible. So here, ideally, you want to test one key assumption and make sure all your parameters are either measurable or can be inferred from data. And for testing, you want to use diverse doses and conditions. And once you do these, you will hopefully build a model that generates unbiased predictions. For an example which opposes all of these rules, you can see this example here. So coming back to the list of questions that we asked, first of all, are PBPK models sloppy? Yes, there are, because lots of parameters. For the drug parameters, such as partition coefficients, they're unknown. And secondly, how can you predict with confidence? Well, keep your model as simple as possible. And on the other hand, test your model with a lot of data. And these data should be generated under diverse conditions. And lastly, which open project is worth doing? Well, in my lab, I keep a few open projects hosted on these URLs. And you do not have to be a modeler to contribute. You can write about it so that more people know about these projects. Thank you very much.